Hello and welcome to G Fun Facts Online, your go-to spot for the coolest science-making headlines. Today, we're diving deep, really deep, into the microscopic world with a technique that's completely changing how we see life itself. We're talking about cryo-electron microscopy, or cryo-EM for short. Imagine having super-powered goggles that let you see the tiniest building blocks of life, proteins, DNA, even viruses, in stunning, almost atom-by-atom detail. That's what cryo-EM is delivering, and it's a total game-changer for science. So what's the secret sauce behind cryo-EM? At its heart, it's pretty ingenious. Scientists take biological samples, like those little proteins or viruses, and flash freeze them super fast. We're talking colder than -150 degrees Celsius, usually with liquid ethane. This isn't your average freezer burn. It creates a super thin layer of vitreous ice, which is like glass, non-crystalline. The amazing part? This super fast freeze locks the molecules in their natural, active shapes, just as they are in our bodies. This is a huge deal because other methods, like X-ray crystallography, need molecules to be crystallized, which can sometimes change their structure. Cryo-EM says, nope, we want you au naturel. Once our samples are perfectly frozen in time, they go under a powerful transmission electron microscope. This microscope doesn't use light. It fires a beam of electrons right through the sample. As the electrons pass through, detectors capture thousands of 2D pictures, like tiny shadow puppets, from all different angles. Then comes the really clever bit. Powerful computers and super smart algorithms take all these flat 2D images and reconstruct a full 3D model of the molecule. And we're not talking a blurry blob. These 3D models can show us the precise arrangement of atoms. Now, cryo-EM isn't brand new, but the advancements in just the last few years? Absolutely mind-blowing. It's like going from an old blurry TV to a super ultra HD 8K screen overnight. First up, the eyes of the microscopes got a serious upgrade. New direct electron detectors, or DEDs, are way more sensitive and faster. Think of them as super high-speed, high-resolution cameras for electrons. This means clearer images with less noise. Plus, better electron sources and fancy bits like phase plates have boosted contrast and resolution, especially for molecules that were previously too small or tricky to see clearly. Then there's the brain power, the image processing. We're talking seriously sophisticated software. Scientists are using advanced algorithms, even incorporating Bayesian statistics and deep learning, yes, AI, to sift through mountains of data. This helps them pick out the individual molecules from the images, deal with the fact that molecules can be a bit flexible and exist in slightly different shapes, and build those incredible 3D models. Software like Relion and CryoSpark have become the rock stars of the cryo-EM world. And speed? Oh yeah, we're talking warp speed compared to the old days. Automation has swept through the whole process, from preparing samples to collecting data and processing images. Structures that might have taken years to figure out can now sometimes be solved in weeks or even days. This is massive for things like structural genomics, where scientists want to map out the structures of thousands of different proteins. Because of all this, cryo-EM is tackling stuff it couldn't before. It's not just for giant, stable molecular complexes anymore. Scientists are now getting amazing views of smaller proteins, tricky membrane proteins, which are embedded in our cell walls and notoriously hard to study, and dynamic, flexible molecular machines caught in the act. There's even a cool variation called cryo-electron tomography, or cryo-ET, which lets us see molecules in 3D right inside their natural home, the cell. Talk about a window into life. And get this, the resolution has become so good that for some well-behaved samples, we're talking true atomic resolution, around 1.2 angstroms. That's detailed enough to see individual hydrogen atoms. This level of detail is crucial for understanding exactly how molecules work and how drugs can interact with them. So what does all this incredible molecular peeping mean for us? The impact is huge. In drug discovery, cryo-EM is a revolution. By seeing the exact 3D shapes of proteins involved in diseases and how potential drug molecules bind to them, scientists can design much better, more targeted medicines. It's also helping us understand how drug resistance happens, which is a massive challenge in medicine. It's also unlocking the secrets of fundamental biological processes. We're getting an unprecedented look at the intricate machinery inside our cells that handles things like DNA replication, how our genes are read, how proteins are built, and how cells talk to each other. Seeing these machines in different functional states helps us understand exactly how they operate. Cryo-EM is also a great team player. It's often used with other techniques like X-ray crystallography and NMR spectroscopy, plus computer simulations, to build an even more complete picture of how molecules look and behave. Looking ahead, the cryo-EM story is far from over. Scientists are pushing for even better resolution, trying to capture molecular processes as they happen in real time, like making molecular movies. There is also a big push to develop smaller, more affordable cryo-EM instruments so more researchers can get access to this amazing tech. And it's even branching out beyond biology into materials science, helping to image delicate materials that are sensitive to electron beams. In a nutshell, advanced cryo-electron microscopy is giving us an ever clearer, more breathtaking view of the machinery of life. By visualizing the building blocks of biology in their natural state at incredible detail, it's driving forward our understanding of life itself and opening up exciting new paths for medicine and discovery. That's all the time we have for today on G Fun Facts Online. Thanks for tuning in to explore the amazing world of cryo-EM. Until next time, stay curious.